Hi, Mr. Wartsky here, and today is the third screencast and the last screencast of protein synthesis. Yay! So the consequences of mutations deal directly with the synthesis of proteins because shape is everything. So if we have the original DNA over here and the base substitution down here, in class, you're going to be asked to give me the mRNA, the tRNA, and the amino acid sequence of this sequence. So this is the original DNA here. You're going to give me all three of those things. Write that down. Then underneath it, write down the mRNA, and write down tRNA and DNA. Remember, amino acids. Remember, that you get if you use the mRNA codon table, you're going to use the mRNA to get your amino acids. The base substitution. So find the mutation here. Right here, I'll give you a hint. Right here. Right, we change it from an A to a G. How do we do that? We're comparing these things here. That's all we did. We can change it from an A to a G and write down the mRNA, the tRNA, and the amino acid and tell me if it's harmful or not. Okay, beautiful. Now, probably the most harmful mutations are the frame shifts. So when we add something like an insertion, we shift the frame, we shift the codons down. If we delete it, we shift the codons down the other way. So in this case here, instead of having 3 T's, we have 2 T's, right? We're comparing back to the original. Over here, where's the mutation? Oh, we added a T here. We added a T here, and that shifted everything down. So write down the mRNA, the tRNA, and the amino acids, and see what that type of mutation does to the protein sequence. It probably plays havoc. Over here, we're taking 1 T away from here, and what if we removed anything else? No, I think that's it, right? Just we took our T away. Okay. So again, write down the mRNA, the tRNA, and the amino acid. Here's the original. Again, the inversion, we flipped a section. So what section did we flip? That usually happens during crossing over. I believe right here. We flip this internal section. The the C is the same. The 2 C's are the same here and here. Or maybe they're different. Yep, we flipped everything from here down. Flip. Breakage, we cut the DNA sequence off there. That obviously has a huge role to play in protein synthesis. And then we have extra copies. Boom. Okay, if you find the extra copies, you'll be all set. Now, there are polyglutamine diseases, which are extra CAG sequences, and specifically found in Huntington chorea. So if you have 39 CAGs, you will have Huntington chorea by 66. If you have 40 CAGs, Huntington chorea by 59. If you have 50 CAGs, you'll have Huntington chorea by 27. And Huntington chorea, the protein shape is so misshaped that it will collect in your brain cells and eventually kill your brain cells. So in Huntington chorea, you lose brain cells and you die after a period of time. And we'll talk about Huntington chorea later on when we talk about genetic anomalies. But the worst mutation are called frame shifts. Yep, those are your deletions and insertions. They shift everything up or down. Now, there are 3 categories of mutations. You can have harmful, harmless, or beneficial. Harmful is like if you blindfold yourself and I were to shut off your engine of your car, put you in front of the car, lift up the hood, have you reach inside there and do something to your car, grab something, and most likely it'll be harmful. So it's going to be hurtful. It's going to harm the organism if you have a mutation. That's most of the time. Sometimes it can be harmless, right? You can change like base substitutions. You can change it from one base to another and still keep the same amino acid or still keep the same type of amino acid so you maintain the protein shape. And rarely will a mutation be beneficial, but it can happen. For example, the masseter gene codes for the masseter muscle. The masseter muscle runs from the top of your skull all the way down your face to your jaw and allows you to close your jaw. Few people have a weak masseter compared to the other hominids or other organisms that look like us, like apes or chimpanzees. Right? So it is 4 times weaker than the gorilla. We compare the gene sequence of human people masseters to gorilla masseters, we find that there are 2 deletions. 2 deletions. It's amazing, right? So the 2 deletions create a weaker masseter muscle. But that also allowed for our skull to expand and for our brain to get bigger and for us to have thoughts so that we can figure out that the 2 mutations in the master gene in gorillas was beneficial to us. Amazing. So it is possible to have beneficial mutations. And that is it for the mutation presentation. If you have any questions, please ask me tomorrow in class or refer to your book or listen to the video over again. It's only about 5.5 minutes long. Now remember, in class, we're going to go over the consequences of mutations. So you should be able to give me the mRNA, the tRNA, and the amino acid sequence for the things that I put in the screencast. Have a good night.